Okay, Dr. Landry has selected our next 2024 Canada Gairdner Early Career Investigator, who is Dr. Raquel Cuella Martin. Dr. Martin joined the Department of Human Genetics at McGill University as an assistant professor in 2022. She undertook her PhD studies at Dr. Chapman's lab at the Wellcome Centre for Human Genetics at the University of Oxford, and during that time described the mechanistic role of the DNA repair protein 53BP1 in optimal p53 tumor suppressant responses. Her postdoctoral work as an EMBO long-term fellow at Columbia University used CRISPR-dependent base editing to perform genetic screens at nucleotide resolution and functionalize DNA variants at scale. And this approach allowed her to identify loss, gain, and separation of function mutations and new functional domains in DNA damage response proteins, and to functionalize variants of uncertain significance in cancer predisposition syndromes. Now her lab applies cutting-edge, large-scale precision genome editing to approach complex biological questions and to improve intelligent drug design. Dr. Cuella Martin will be presenting Understanding Unique Protein Features with Precision Genome Engineering. Please join me in welcoming her to the stage.
Well, first, thank you, everyone. Thank you to the Gairdner Foundation for this honor, of course, to Dr. Landry. And I also want to extend my thank you to the whole Canadian community, because as a newcomer, I have been welcomed here with open arms. So basically, that introduction said a bit of everything that I'm going to be talking about today. So let's just go into it. So we have heard today a lot about basic science and really fundamental questions, so I'm going to give an example of that during my talk. I have been working on this protein that you see up there, 53BP1, for almost 10 years now, since the beginning of my PhD, so I'm one of those researchers that worked on the same protein for their whole career. And I find it a very interesting protein. So you see it's fairly large. It has a very long N-terminal that is not a structure, so it makes it tricky to study, but uses that as a platform to recruit factors to DNA damage sites. And it does so by forming big oligomers and recognizing two key histone marks as sites of lesions that are H4K20 dimethyl with the tandem Tudor domain and H2AK15 ubiquitination with the ubiquitin-dependent recruitment domain. And all this architecture is essential for the repair of DNA double-strand breaks by a non-homologous end joining. And this process is very important in physiological conditions. We have heard today about the generation of the T-cell receptors, so very important for VDJ recombination, also very important for the generation of the antibody repertoire. But it's also an extremely toxic pathway when we find BRCA1 deficient tumors. This non-homologous end joining drives the generation, as you see there, of these massive radial chromosomes and massive chromosomal rearrangements. And 53BP1 had been studied in this context for a few decades, but we were interested, this is working. We were interested on this extreme C-terminal region of this protein that is called the tandem BRCT domain. And this interacts with, master tumor suppressor P53, and also with the deubiquitinated enzyme USP28, brings them together, and that function is essential for optimal P53 anti-tumorigenesis in response to different lesions, such as genomic instability, but also other groups reported that this is critical when you have extended mitotic division, and your cells could probably missegregate your DNA, and basically this pathway goes and kills those potential cells that will cause problems. So when I was studying this during my PhD, we are molecular biologists. We were using traditional molecular biology techniques. So first, you establish a loss of function model. So we use CRISPR-Cas9. That was just, I guess, just published back in the day to generate genetic knockouts for 53BP1 and also for P53. And we put a drug that is a specific P53 activator. The drug is called NATLIN3. So obviously, when you see there, when you don't have the target, you don't have P53, the cells are fully resistant. And when we didn't have our protein of interest, the cells had an intermediate phenotype. They were also resistant, indicating us that there was a functional relation there. and then to disentangle the mechanisms, because this is what we do in the lab. We are very interested in disentangling the mechanism of how things work. We will complement that phenotype, put back a wild type, 53BP1, and see whether we were able to revert that resistance, but also put back design mutants that we made based on how we thought the protein worked, right? So if you have there, what I said, the tandem tutor domain, that's a methyl binding module, we will mutate the methyl binding pocket. And then see whether that was involved or not in driving that resistance, and then if it was doing so by fostering the formation of the complex with p53 and USP28 that are, the discovered binding partners. But this had a fundamental problem, right? All our structure function that we call it base analysis were biased, were not really systematic. It was based on how we thought the domains were, how we thought the protein worked. And also we had additional problems that prevent us from using demutational scanning and other more systematic approaches, because our proteins were difficult to express ectopically. You see 53BP1 is not a small protein. They were large, highly structured, heavily regulated. So this complex was really telling us, you need to figure something out to study this on the physiological context and in a systematic manner. So then I moved on to my PhD, from my PhD to my postdoc work, and that's what I set out to do. And we devised the implementation of CRISPR-dependent base editing at high throughput. And this is a simple schematic of how one of those experiments look like. So basically, we have our pool of cells that express a base editor. And in the case of the screens I'm showing here, this is a cytidine base editor. So we have the Cas9, a nickase format, fused to a cytidine deaminase that makes C2T transitions in a specific area of the genome where we design our guide RNAs to obviously bring that base editor. And then we bring that base editor to our genes of interest by putting guide RNAs all across the coding region of that gene and making those C2T transitions in a systematic manner. And after that, we obviously put those guide RNAs into the cells. We added p53 activating drugs to enhance those phenotypes. And then using next generation sequencing, we can sequence the end population versus the initial population and figure out which mutations are behaving like loss of function, giving us resistance to the drugs, or potential includes even gain of function, giving us sensitivity to the drug. So if we zoom in one of those maps that we have over here, you see actually that the mutations we have nonsense and splice variants in yellow and red as reference for loss of function. So you can see the mutations cluster in specific regions of the protein. They are not stochastically all across. And we were particularly interested on the mutations place in the tandem tutor and the tandem BRCT domain because we had been studying those domains for quite a while and we thought we had it figured out by now. So those are crystal structures from the tandem BRCT domain. On one side, you can see the results from one of your screens. So in the purple, positive log-fold changes, so cells that were given mutations that were given resistance, loss of function, and then in blue, you have sensitivity. And then side by side, we have amino acid conservation. And what was surprising to us, because we usually attribute to amino acid conservation if a region is conserved. We think that that's functionally relevant or structurally relevant. And when we were seeing where our mutations were localizing, if you look for these dark spots over there, they were not really correlating with areas that were strongly conserved. Right down here, that was not really strongly conserved. But our mutational analysis was really telling us there was something important right there. And the same was the case for the 53BP1 tandem tutor. You have here in the front the methyl peptide binding site. That's the canonical way these domains bind, and it's very strongly conserved. But then all our mutations that we were finding here were mapping right there to the rear interface. And then we had some gain of function, potential gain of function mutations on the other side, but really not in an area of a strong evolutionary conservation. So we were quite puzzled by those results, and we decided to figure it out molecularly. That's what we do best. So we selected the specific mutations that were being made in the tandem tutor domain and validated it one by one in what we call two-color competitive growth assays. So basically, we put cells that are wild type, color here in red, cells with our mutation, color in green, and then we mix them and let them fight, and then see which one overpopulates the other. And basically, this told us, the ones that look like loss of function in our screen are having the exact same phenotype in these one-on-one assays, and the same with the one that look like a gain of function. And then we do deep sequencing at endogenous locus to verify exactly which are the mutations that are being inserted. And in this case, we had valine 144 to isoleucine, glycine 1562 lysine, giving drug resistance, so really loss of function, and G1593 to lysine, giving drug sensitivity. So we always go back to the structure, and you see they are placed right there, where I told you that dark spot was coming up in the structure before, and it's really on the opposite side of the methyl binding module. And so we were thinking, well, this is probably really not required for 53 if you want to go to sites of DNA damage, because unless it's allosterically regulating that, like it's so far apart. And that was really the case. So despite the presence of those mutations, 53BP1 was able to be recruited to double-strand break lesions like a wild-type control. We have the AABS1. And it was actually functional in those sites because it was enabled to restore homologous recombination in BRCA1 deficient cell as deleterious mutations will do. But again, once more, those mutations were critical in p53-dependent responses. We put them in NALIN3. Loss of function mutations were resistant, gain of function mutation was sensitive, indicating that they were regulating p53, and this was perfectly correlating with their ability to form the complex between these three proteins. So both loss of function mutations had trouble binding to USP28, and then the gain of function mutation was actually binding better, so forming probably a better complex and regulating p53 better. So at this stage, we were very happy, and we were like, okay, great, we have figured some more things out. These type of screens, this type of systematic mutagenesis analysis allow us to discover loss of function, gain of function, separation of function mutations, and very interestingly, a novel protein-protein interaction surface that is not the way these domains usually interact, and is like super rare, and we have figured out how this protein binds to USP28. Right, well, that maybe was not really the case, because we had all this knowledge, and when you have that knowledge, sometimes you go to the structural prediction softwares, and this was done with AlphaFold3 multimer, and we're like, okay, now we can probably tell which prediction seems accurate based on our mutations, right? And this is the tandem TDOR domain, now placed in a little bit of a different position. Right over there is the methyl binding pocket, right over here, our pocket of interest, and we place USP28 there. And it was really not binding where we thought it will bind, right? Like, our co-IP was telling us that that mutation was disrupting, but it was really not binding there. And in fact, what was binding there was the tandem BRCT from 53BP1. And it was doing so with that region where we also localized mutations based on our analysis. I was like, okay, well, that was not at all what we were thinking. But if we put P53 right there in the mixture as well, you see that actually where we find mutations with our screens is where those partners are binding. You see there the P53 BRCT interface, and you also see there USP28 having contacts with the BRCT right where our mutations were identified from the screen. So probably, what we were seeing in our biochemical assays were just like the loss of this internal interaction was releasing the whole complex and moving USP28 out. Because both domains in 53BP1 are required to retain the interaction. So not really how we thought they worked in the first place. And of course, we do think these type of tools have a lot of potential for discovery, right? And we are not only interested on those questions, we are interested in most translational questions, so I choose a basic one, but we are also interested in others, such as how to use these systematic mutagenesis to map allosteric sites and find a way to drug them, to identify novel functional domains, even in proteins that we have been studying for many, many years, as it happened here. And also we have some projects on using CRISPR-based technologies to identify targets for rare disorders. And this is, maybe what's going on in the lab, but then we have managed to convince different groups to work with us with these, and we have projects in collaboration ranging from regulation of mRNA transduction by mutating non-coding regions to understanding, maybe appealing more to this audience, TCR antigen interactions. And with that, I would like to finish by saying thank you to my lab members and also to my PhD and my postdoc lab that I saw some working here, and to all our collaborators. And also, I would like to also say thank you to all the labs that we have managed to convince so far to join our base editing screening crew, and they believe that this tool is useful for their research as well. And I think that that's the beauty of it, like when you have a tool that you think is useful for research across disease areas, share it, encourage the usage, and try to see to do that research all together. Thank you everyone.